Every time you see a genetics passage on the MCAT, there's one question hiding underneath it. Where does all this variation actually come from? Meiosis isn't just cell division, it's a diversity engine. In this video, we'll break down the key mechanisms like crossing over, independent assortment, and random fertilization, and connect them directly to the kinds of passage-based questions you'll face on test day, so you can recognize them instantly and answer with confidence. Meiosis consists of two sequential divisions that reduce chromosome number from diploid to haploid while producing genetically unique gametes. In Meiosis 1 and Meiosis 2, a single round of DNA replication is followed by 2 rounds of division, ultimately yielding 4 haploid cells from 1 diploid precursor. The key distinction from mitosis is that homologous chromosomes, not sister chromatids, separate during Meiosis 1, which is called the reductional division because it halves the chromosome number. Prophase 1 and subdivided into stages where homologs pair, or synapse, forming tetrads, and exchange genetic material. Metaphase 1 aligns homologous pairs at the equator, anaphase 1 separates them, and telophase 1 produces 2 haploid cells that still have duplicated chromosomes. Meiosis 2 resembles mitosis, where sister chromatids separate, leading to 4 genetically distinct haploid gametes. A helpful mnemonic is PMAT twice, but remember that the big conceptual difference is in Meiosis 1, where homologs separate. On the MCAT, you are often tested on ploidy changes, the difference between homologs and sister chromatids, and identifying stages based on chromosome behavior. Expect passage questions where you must track chromosome number or DNA content, such as distinguishing 2N from N or duplicated versus unduplicated chromosomes. Crossing over during prophase creates genetic recombination by exchanging DNA between homologous chromosomes. During synapsis in prophase 1, homologous chromosomes align tightly along their lengths, forming tetrads composed of 4 chromatids. At specific points called chiasmata, non-sister chromatids break and rejoin, swapping segments of DNA. This process generates new combinations of alleles on each chromosome, increasing genetic diversity beyond what either parent originally had. A useful way to remember this is that crossing over literally crosses genetic information between homologs. Mechanistically, enzymes create double-strand breaks and repair them using the homolog as a template, ensuring reciprocal exchange. On the MCAT, you may see questions about recombinant versus parental gametes, or gene mapping, where recombination frequency reflects physical distance between genes. The farther apart 2 genes are on a chromosome, the more likely crossing over will occur between them. This connects to genetics topics like linkage and recombination frequencies, often tested with Punnett squares or pedigree-style reasoning. Independent assortment during metaphase generates variability by randomly distributing homologous chromosome pairs into gametes. Each pair of homologous chromosomes aligns independently of every other pair at the metaphase plate, meaning the orientation of one pair does not influence another. As a result, maternal and paternal chromosomes are mixed in different combinations when they separate in anaphase 1. The number of possible combinations is 2 to the power of n, where n is the number of homologous pairs. In humans, with 23 pairs, this results in over 8 million possible combinations even before considering crossing over. A helpful mnemonic is think of each chromosome pair as a coin flip, heads or tails, maternal or paternal. On the MCAT, this concept often appears in probability-based genetics questions, where you must calculate the likelihood of specific allele combinations or understand how variation arises even without mutation. This also ties into Mendel's law of independent assortment, though that law strictly applies to genes on different chromosomes or far apart on the same chromosome. Errors in meiosis, such as non-disjunction, can lead to aneuploidy, affecting genetic variability and causing disorders. Non-disjunction occurs when homologous chromosomes fail to separate in meiosis I, or when sister chromatids fail to separate in meiosis II, resulting in gametes with abnormal chromosome numbers. When such gametes are fertilized, the resulting zygote may be trisomic or monosomic. Classic examples include trisomy 21, which causes Down syndrome, as well as Turner syndrome, where there is only one X chromosome. A useful way to remember this is that non-disjunction equals no separation. The stage matters. If it occurs in meiosis I, all resulting gametes are abnormal. If in meiosis II, only half are affected. On the MCAT, you may be asked to determine which stage the error occurred based on the pattern of gametes or offspring, or to predict outcomes of fertilization involving abnormal gametes. This concept also connects to cell cycle checkpoints and chromosomal segregation mechanisms, reinforcing how tightly regulated meiosis must be. Genetic variability in meiosis is further enhanced by random fertilization, combining diverse gametes from two parents. While meiosis generates diversity through crossing over and independent assortment, the fusion of any one sperm with any one egg multiplies this diversity dramatically. In humans, each parent can produce over 8 million distinct gametes due to independent assortment alone, and when you consider fertilization, the number of possible zygotes exceeds 64 trillion combinations, not even accounting for recombination. This means that each offspring is genetically unique, except in the case of identical twins. A helpful way to conceptualize this is that meiosis shuffles the deck, and fertilization deals a completely new hand. On the MCAT, this concept often appears in questions about sources of genetic variation, where you must distinguish between mutation, recombination, and fertilization. It also connects to evolutionary biology, as genetic diversity is the raw material for natural selection, reinforcing why meiosis is essential not just for reproduction, but for population-level adaptation.